Hello, Harry Brook here, and we're talking to Syama Chatterton, who is the pulse crop pathologist with Agriculture and Agri-Food Canada in Lethbridge. And today we're going to be talking about everybody's favorite, pulse diseases.
You're listening to the official podcast of Flagstaff County.
Good morning, Syama.
Good morning, Harry.
I'd like to, start with one of the things I've heard over the years with producers is when they first try pulses or peas, mostly is the first thing they'll try. They say, had a really good crop. It was really great the first year. And then for some reason or other, yields never made that same level. And I'm just sort of wondering what possible causes could be the issue there that makes it limits the yield potential on those pea crops.
Yeah, that's a really good question. And I think what it all comes down to is our favorite topic of pulses, and that is root rot diseases. Root rots are caused by a couple of different pathogens. The number one pathogen being Aphanomyces euteiches. I know that one's quite a mouthful. And then also what we have associated with Aphanomyces is Fusarium species. There's a huge range of different Fusarium species that are associated with pulse crops.
And other crops too.
And other crops as well. Yes, that one, we have different species, different crops. Some of them go across all crops. It's a very complicated group. And so what we find is that every time you grow a pulse crop, you tend to increase the level of these soil-borne pathogens. So even Fusarium species, some of them might start out low, and then every time you grow a pulse crop, you can increase the presence in the soil. And then particularly that's what we see with Aphanomyces. It has been found at very, very low levels in kind of native pastures, tame hay pastures that have never been cropped to pulses.
Oh yeah.
And then every time you add a pea or a lentil, you're kind of boosting that level in the soil until you hit your threshold. So
what characteristics of those diseases cause it to sort of hang around for so long and sort of build up those levels? Because, a lot of times we say guys will have a rotation where it's pulses every 4 years. Is that an adequate rotation to keep the levels down to a manageable level?
Yes. So with Aphanomyces, the reason why we call it our big bad, or the one that's the most concerning, is because it has these very long-lived resting spores. So every time, if you have a field that has maybe some really low levels of Aphanomyces to start with, and then you plant your pea crop, you might get a little bit of disease that you don't even notice. And then it leaves behind these resting spores that are called oospores, and those can survive in the soil for a long time. And so then every time you're putting a pea or lentil crop, you might be just incrementally increasing those oospore levels until you get to that threshold where you start to see disease. And we haven't quite figured out how many cropping cycles of a pea or a lentil it might take you to hit threshold.
Okay.
That seems to be, I don't know, sometimes I just say it's luck.
Or is it just more complicated because it's not one single factor, right? It's a multiplicity of factors.
Exactly. It's, if you crop pea or lentil and it was a really dry year or a drier year, you won't get as many oospores returned to the soil. If you happen to, every time you planted a pea or lentil in that field, you hit a really wet year, then you're really going to get a lot of oospores returned to the soil. So it's a combination of what the environment was when you grew your pea or lentil crop. Even the soil type has a big impact on that. We're looking at whether crops in rotation maybe even have an impact on that.
Okay, because even non-pulse crops can affect the liveliness of, or I guess the numbers of infectious.
Yes, that's one of the questions or hypothesis that, if there's some crops that will actually help reduce the oospore load in your soil. So we're looking at things like mustard and oats, even fava beans, because these can all potentially act as what would you maybe call trap crops, where they might induce these oospores that are sitting there. They might induce them to germinate, but they can't actually infect these crops very well. So you might see some reduction in your soil, but it's such a complicated disease that we've been looking at some of this research, but every location we kind of get a different result. So it's hard to come up with really clear answers. And that different result is really because it's so dependent on soil type and environment.
It's kind of a case where, you know, you'd like things to be black and white, but they're usually multiple shades of gray, right?
Yes, that's
and exactly it's more complicated. So talking about the longevity of those oospores, how long can they live? Do you know sort of how, I guess, the viability, or is there any drop-off point?
Yeah, that's another really good question. Again, hard to study because it might take a long time. And the one paper that we sort of look at actually came out in the early 90s, where they tried to kind of measure changes. And they sort of said, okay, an oospore mortality rate of maybe between 30 to 50% per year. You know, so if you're looking at the half-life, you're like, okay, well, how many years does it take to kill half of those oospores? So, you know, that would give it a half-life of 1 to 2 years, depending if you go with the 30% or the 50%. So it can take a long time. You know, if you're up at threshold, above 100 oospores per gram of soil is what we would call threshold. And most soils that we're looking at that are heavily infested are about 1,000 oospores per gram. You know, it can take 6 to 8 years just with natural decay of these oospores to kind of get them back below threshold. We have just started some experiments where we are trying to, you know, really drill down to exactly, okay, what is this mortality rate in the soil? So actually just this fall in November, we went and buried a whole bunch of oospores, and then we're just going to pull them up every 3 months. Yeah. to dig out some oospores in February in frozen ground, and then just, you know, see what happens. Yeah.
I did some work once with an animal insect researcher, and we were burying fly larvae in manure. Horn flies. That was fun. So much fun.
Yes. Yeah.
But I guess, how close is the parallel between the oospores of Aphanomyces and the oospores of clubroot, which would be, you know, it seems to me they're almost equated relatively.
Yeah, little bit of a difference, and I'm not a clubroot biologist, so hopefully I don't get my understanding wrong. So the oospores are very, very thick-walled, kind of single cells.
Yeah.
Whereas my understanding of the clubroot resting spore is you kind of get the clubs, I guess, that are filled with resting spores that are not as resilient as the oospores. So I think with clubroot, you actually can get a decline of those resting spores a lot faster. And then the other difference is that, you know, when we're talking about oospores, our threshold is saying about 100 oospores per gram of soil. Whereas for clubroot, the threshold is usually much higher.
Yeah. Right.
Yeah. And it's because oospores, I mean, clubroot does the same thing too. They have these 2 phases of their life cycle. Mm-hmm. So the oospores are what's challenging. They're what sit in the soil, sit there and wait for their favorite meal. But once that favorite meal is there, like a pea or a lentil crop, then the oospores germinate to produce a secondary structure, which are called zoospores.
Right.
And one oospore can produce like 1,000 zoospores. And then it's the little zoospores that swim to the roots and actually infect, right? So 100 oospores actually can mean 10 to 100,000 zoospores are there. And
interesting. Yeah. I didn't know that.
Yeah.
I always think it's sort of cute. Well, not cute actually, but, you know, got these little flagella zoospores, you know, it's kind of like they're targeting almost like, I was thinking of cruise missiles.
Yes.
And they sort of just
That's
swim through it. So dry
That's-
years.
yeah, that's exactly it. And like you said, they need
down.
moisture to be
Yeah.
able to, with their little flagella, to be able to swim. And we talk about swimming, but they're not like marathon Olympic swimmers for them. It's it's a couple millimeters, sorry, I said meters, couple millimeters
Yeah.
actually that they can swim, but to them that's a long way, but it's why they need moisture and these like soil, the pores in the soil kind of have to be full in
Yeah.
order for them to swim through them to find the roots. So that's why environment is such a huge part of this disease. Yeah.
So even what causes them to germinate? Would it be the root exudates from peas?
Yes, that's exactly it. It's the root
Okay.
exudates from pea or lentil. Some unknown, actually we have another research project on that right now that was just
Cool.
funded by Alberta Pulse Growers and Ardar. What we're looking at is, okay, if we can target exactly what that compound is that's coming from the pea, can we then figure out, take advantage of that somehow to disrupt that life cycle, if we can trick the oospores to germinating when there's not a pea there or something like that, right? So yeah,
and blow their wad all in one go.
Yeah,
so that,
exactly.
reduce the numbers.
And they're like, surprise, it's actually a cereal crop in here or something like that. But so we are looking at that. And then I think what's interesting, why maybe fava bean has some impact, because it appears that oospores will germinate after kind of from fava bean root exudates, but they can't actually infect the fava beans that well. There's something going on there with some sort of resistance to fava bean. So there's researcher at AFC in Saskatoon that's actually trying to look at that kind of differential fava bean response. Yeah.
Okay, so I used to be a DA a long time ago. We're talking decades. But I remember with pulses, it was sort of like you sort of have a stage of growth when they started dying off, sort of told you what particular disease was causing the problem. So if it was early, it was, I don't know, maybe a common root rot or something. And then later on, it was fusarium. And then as she's flowering and dying, then it's Aphanomyces. At least that was kind of the rule of thumb that we were taught.
Right. Yeah.
I don't know if that still holds.
Yeah, that's a really good question. I think what we would see here is often the opposite. If it comes on early in the season, after there's been, okay, I should back up a bit. If it's been a wet spring, you will notice that your plants might start yellowing mid-June. And that's actually the best time to see Aphanomyces on the roots in its sort of pure form. It's when the roots display the best symptoms of Aphanomyces.
Oh, okay.
Then if you start checking your roots maybe later July into flowering and into July, it's when we start seeing a lot of fusarium colonization. So then you might pull up your roots and really not know what's going on, because
okay, but it's secondary then.
That's where it's complicated. It can be secondary, but fusarium root rot can also cause disease
by
on
itself.
Its own.
Yes.
Yeah. But what we found by doing greenhouse studies is that when you have Aphanomyces and fusarium present, it's when you have the worst disease and the biggest impact. Yeah.
Okay, so with fusarium, I was told that if you cut it off at the root, you should see some reddish in the stem, which is a sign of Lisa fusarium, right?
Yes. Yeah, that's a pretty good symptom that there's fusarium there. It does move into the vascular system, and then you get this, and in culture, fusarium is kind of a bright pink magenta. And so you'll see that in the root when you cut it open, that you have this kind of bright pink. It's a really pretty color, but not so pretty with your end result of view. Yes. Yeah.
What about irrigation? Is that a problem with peas and irrigation, or pulses and irrigation?
Yes. And we don't have a huge irrigated pea crop in the prairies, but there is some definitely down here in the southern Alberta region. There's peas that are grown under irrigation, particularly for processing peas, for green peas.
Mhm.
Yes, we do see that it can be more of an issue under irrigation because, that moisture is always there, and that moisture that they love. So even like a dry year, 2021, you still see it being a problem because moisture's there.
I was once told that pulses are one of our most water use efficient crops. Is that true?
I believe that is true. Yes, based on data that I've seen. I'm not a water use specialist, but just based on some of the tables and charts that I've seen, they are very efficient users of water. And when you look at a pulse root, and this could also be why we have so much trouble with root rots, they don't have this really like abundant root system that you would get with like a cereal or even canola. It's actually quite easy to dig up a pea or lentil root, and they really kind of stay on that top layer, the top 12 centimeter, sorry,
The
20
vast majority
centimeters.
of the roots
are kind of right in that top layer, right? So, how that makes them efficient water users, couldn't tell you exactly how that plays into it, but they just, oh, that. Yeah.
Well, I don't know if I know it, but I've been reading up on AM fungi, right? The arbuscular mycorrhizal fungi.
Yeah.
And they were saying one of the benefits of them is that the hypha they're a lot narrower than the root hairs. And so they're able actually to access water that is not available to the root hairs.
Yes.
So, and if that's the truth, that would kind of explain some of the water use efficiency.
Yes. Yeah, I think so. And I know that pulse crops, there's different crops are better at kind of recruiting mycorrhizal fungi and that association. And I think pulse crops have a very good association with mycorrhizal fungi. So
the whole thing of like the AM fungi and I guess what you call, I hate using the word soil health, but I find it fascinating because it gets really, really complicated.
Yes.
And basically each individual field and soil is an entity unto its own, I think.
Yes, for sure.
Yeah.
And, this whole issue with root rot, we often also think about it in context of soil health.
Mm-hmm.
And, you're like, kind of soil that's healthy under what a soil scientist might have for a definition of a healthy soil. Like if it's supporting these soil-borne pathogens that seem to increase as you grow pulse crops, which are supposed to be really good for soil health.
Yeah.
Then what's that balance between, really good soil health by a soil scientist definition versus a pathologist's definition? Right? So, yeah.
So is there any breeding going on right now to try and find any kind of resistance? Well, like or trying to maybe transfer some of the genes from the faba beans into peas, possibly?
Yeah, so I guess I'll answer the first question about breeding for resistance. There is breeding going on. And there was a huge concerted effort, in the 90s, early 2000s from groups in France and the US that were dealing with Aphanomyces much earlier than we were here in Canada, to really try to discover and develop pea lines that had partial resistance. So those were released as lines, I think in about 2012, 2013. So there is work with those lines to try to transfer that partial resistance from pea into agronomically elite cultivars, right? Part of the issue is that some of those lines that have resistance don't have the good agronomic traits that we want. They're still fully leafed. Sometimes they have, the colored seed coat. They're not the yellow or green cotyledons, and so that it can be really hard to break that linkage between kind of the colored flowers and colored seed coat and resistance.
Mm-hmm.
So that's one of the challenges. And then I think, to be able to transfer resistance from something to faba bean to pea doesn't really work because you can't cross them. They're too far distance related.
The genetic, Yeah,
but it's like, okay, well, if we can discover what's resistant in faba beans, if there's some genes that are responsible for resistance, can we start to look for those genes in peas or use marker assisted selection to, like I said, look specifically for those traits in peas or some of the more the wild relatives of peas that you can actually cross with pea. So learning what some of this resistance is in other pulse crops, I think, can be very important for developing resistance, but it's the more long-term track, right? But yeah.
What about seed treatments? Is there, because I know it's very complicated, because seed treatments, got fungicides on them all.
Yes.
And that's got to affect the populations, but obviously it doesn't seem to affect Aphanomyces much, does it?
Yeah, and that's because Aphanomyces isn't a fungus, just to make things more complicated.
Well, and it's not a bacteria, is it? Sort of like amoeboid,
it's
or
what we call it oomycete. So oomycetes are a group that are often confused to be fungi because they have very similar life cycle habitat as fungi, but they're actually in a completely different kingdom that's more closely related to plants, actually to algae, than it is to fungi. They just happen to, convergent evolution, as they call it. They just happen to have a very similar lifestyle, so they get confused. So because they're not actually fungus, then a lot of the fungicides don't work against them. The other group that people would be really familiar with as oomycetes are pythiums, and Phytophthora actually, which are also plant pathogens. And so there are seed treatments for that. Metalaxyl is often included in these seed treatment packages, but metalaxyl has never had activity against Aphanomyces for whatever reason.
What a pity.
Yeah. And so then there's a couple of other seed treatments that have activity against oomycetes that have been looked at. And what we find is that you'll get kind of an early season disease suppression if we were more worried about Aphanomyces' effect on, say, germination and establishment of seeds, then it would be effective. But because Aphanomyces does, like I said, it's kind of that mid-June.
It delays a little bit, doesn't it?
Yeah. So
after seed treatment's gone.
Yeah. The seed treatment's gone by the time Aphanomyces becomes active. I think in some warmer climates, Aphanomyces can cause more of like a seedling blight, and then then it would be an effective seed treatment. But because our climate's a bit cooler, Aphanomyces likes a bit warmer temperatures, it doesn't become active until kind of mid-June when our soil temperatures are warm enough. Yeah.
Sometimes I get the feeling that no matter what we grow and whatever we put in the soil, somewhere in there is some pathogen that's going to attack it, no matter what we're using,
Yes, I know.
Kind of plug a hole over here and then the dike springs a leak over there.
Yes.
Yeah.
And like soil-borne pathogens are definitely the trickiest to deal with. The soil is still kind of a big black box. So we don't fully understand how community dynamics even work in the soil. And we don't know, it's very hard to work with. So you can't always test what's in your soil. So it definitely makes it challenging.
It's a challenge.
And it's a lot more harder.
Well, think of it this way. You basically have a lifetime career ahead of you to figure stuff out.
Yes.
And it doesn't look like there's any end to it.
Unfortunately not. I mean, I feel like, it's been about 10 years since we've been, know that this is our problem and been dealing with it. And, we're chipping away at it, but I would say we tried a lot of things. There's a lot of research going on, and, most of it doesn't work. So it's not as simple to, we could just.
It's valuable, right?
Yes. Yeah.
Failure is an extremely valuable process because it leads you to the successful answer.
Exactly. And we're gaining.
Might be like Edison, thousands of failures. Let's hope not, but
no.
So what advice would you give somebody who likes to grow pulses and wants to have some kind of sustainable system in place as far as a rotation? What would you suggest?
What I think is really important is one, keep records. And sometimes I know it's challenging and it's easy for me to sit here and be like, do this and do that. But I know as a farmer, you're juggling so many things. How can you do everything? But I think keeping a record of the environment that was grown when you grew that pea and lentil crop, so that if it was wet or dry. We're trying to come up with sort of these models. Okay, well, how many oospores are left behind in a wet versus dry year? So you would know that. The other thing that I would recommend is checking your roots often. Like every time you grow a pea and lentil crop, make sure that you check those roots, because the shoots can look really healthy, but you could be having to start developing some problems in those roots before it shows up in the shoots. And I would even go so far as to say, every time you have a pea or lentil crop in the ground, collect some roots and get them tested for Phanomyces, because it could be there before you see it, right?
Right.
And then if you have it, say you plant peas in 2022 and everything looks really good, and then 4 years later you put a pea crop in and it could be a wreck, and it could be because you were just at that threshold level in your pea crop in 2022 and didn't see that it was there. So if you get in the habit of checking your roots for Phanomyces every time you grow a pea or lentil crop, you'll be able to tell if those levels are starting to increase in your soil.
Okay.
Yeah, and the reason why I say check your roots and not your soil is because this is a whole other issue. I've been working on trying to develop a soil quantification test for about 5 years. And it's not always reliable, whereas testing from the roots is always reliable.
The evidence is right there, right?
Yes, yeah.
It's much easier to identify it when the disease is present.
Exactly, yes, yeah.
Okay, thank you. Is there anything I've missed about it? Or what let me put it this way. Is there anything more you'd like to add in our little podcast here?
I think we've really talked about a lot of the ins and outs. I mean, the only thing, we always talk a lot about Phanomyces, and I said it is the big bad, but just, be aware that Fusarium root rot can be a problem all on its own. It's not necessarily a Phanomyces if you have root rot. It's just that the fields that have a Phanomyces are the ones that tend to do a lot worse, right? Where you could get away with some Fusarium root rot without having a huge impact on, or as much of a visual impact on yield, but Fusarium root rot could be there just kind of chipping away at your yield, which is why you say, when you opened it up with saying, oh, in the first year it does really well, and then it seems to decline every year. And that could also be just because of Fusarium root rot. That just is it's just like feeding on the roots a little bit. So every year you're, getting a
it's
bit
kind
of
of like growing a baby into an adult, right?
Yes, yeah.
Oh, we now have a monster in the field.
Yeah.
Yeah. Okay, well, thank you, Syama. I've really enjoyed this conversation. And well, if anybody had other questions, could they contact you by email?
Yes, absolutely. Contact me by email. I'm also on Twitter. I think my Twitter handle is at Syama Chat.
Okay.
So yeah, either of those are good options.
All right. Well, thank you very much. It's been a pleasure.
Thanks. It was great to talk to you as well.